students, welcome to the PD Life Science Academy for the Happy Learning. NEET एक्झामचे प्रिपरेशन जे विद्यार्थी करत आहेत त्यांच्यासाठी बेसिक कन्सेप्ट लर्निंग आणि लिंकिंग ऑफ कन्सेप्ट हे जेवढे गरजेचे आहे तेवढेच NCERT बुकमधील चॅप्टरचे कंटेंट रीड करणे आवश्यक आहे. हे करत असताना फक्त एक रीडिंग करून चालणार नाही, मल्टिपल टाइम्स रिव्हिजन होणे गरजेचे आहे. यात टाळाटाळ करणे चांगले नव्हे. आपण सेट केलेले टार्गेट अचीव करण्यासाठी PD लाईफ सायन्स अकॅडमीने सर्व स्टुडंटसाठी जे नीट एक्झामचे प्रिपरेशन करीत आहेत त्यांच्यासाठी 21 डे रीडिंग चॅलेंजची सुरुवात केली आहे. रोज एका चॅप्टरचे रीडिंग आपण कंप्लीट करणार आहोत. हे चॅलेंज ज्या विद्यार्थ्यांनी ॲक्सेप्ट केले आहे त्यांनी त्यांचे डेली टार्गेट अचीव केल्यावर कॉमेंट करून सांगायचे आहे. सो लेट्स गेट रेडी फॉर टेकिंग रीडिंग चॅलेंज. ऑल द व्हेरी बेस्ट. थँक यू. 21 डे बायोलॉजी रीडिंग चॅलेंज. Day 15 reading challenge, chapter number 15 from NCERT biology textbook of 11th standard. That name of chapter is the plant growth and the development. We have already studied the organization of flowering plant in chapter number 5. Have you ever thought about where and how the structures like the root, stem, leaves, flowers, fruits and seed rises, and that too, and by now aware of the terms like the seeds, seedling, plant-led, mature plant, etc. You have also seen that the tree continue to increase in heights or a girth over a period of time. However, the leaves, flowers and fruits of the same tree are not only have a limited dimensions. Flowers and fruits of the same tree are not only have the limiting dimensions but also appear and fall periodically, the sometime repeatedly. Why does the vegetative phase precede the flowering in a plant? All plant organs are made up of the variety of tissues. Is there any relationship between the structure of a cell or a tissue, an organ and the function they perform? Can the structure and the function of these be altered? All cells of the plants are descendant of the zygote. The question is then why and how do they have the different structural and the functional attributes? Development is the sum of the total processes like the growth and the differentiation. To begin with, it is essential and sufficient to know that the development of the mature plant from the zygote, that is the fertilized egg, follow a precise and highly ordered succession of events. During this process, a complex body organization is formed that produces the root, leaves, branches, flowers, fruits and the seeds, and eventually they die. The first step is the process of the plant growth is a seed germination. The seed germination when favorable conditions for the growth exist in the environment. In absence of such favorable conditions, the seeds do not germinate and goes into the period of the suspended growth or the rest. Once the favorable condition return, the seed resume the metabolic activities and the growth take place. In this chapter, you shall also study the sum of the factors which govern and control these developmental processes. These factors are both intrinsic, that is the internal, and the extrinsic, that is the external to the plants. The 15.1 that is the growth. Growth is regarded as one of the most fundamental and the conspicuous characteristic of the living being. What is the growth? Growth can be defined as an irreversible, permanent increase in the size of the organ and its parts or even of the individual cell. Generally, the growth is accompanied by the metabolic processes, both anabolic and the catabolic, that occurs at the expense of the energy. Therefore, for example, the expansion of the leaf is a growth. How would you describe the swelling of the pieces of the wood and when placed in the water? Now the 15.1.1 is the Plant growth generally is the indeterminate. The plant growth is unique because the plant retain the capacity of the unlimited growth throughout their life. The ability of the plant is due to the presence of the meristem at a certain location in their body. The cell of such a meristem have the capacity to divide and self-perpetuate the product, and however soon loses the capacity to divide, and such a cell make up the plant body. This forms the growth when in a new cell is always being added to the plant body by the activity of meristem is called as a open form of the growth. What would happen if the meristem ceases to divide, and does this ever happen? In chapter number 6 you have studied about the root apical meristem and the shoot apical meristem. You know that they are the responsible for the primary growth of the plants and principally the contributes to the elongation of the plants along their axis. You also know that the dicotyledonous plant and the gymnosperm, the lateral meristem, vascular cambium and the cork cambium appears later in life. These are the meristems that causes the increase in the girth of the organ in which they are active. This is known as a secondary growth of the plants. Now the 15.1.2 is a growth is a measurable growth at a cellular level, principally a consequence of the increase in amount of the protoplasm. Since increase in the protoplasm is difficult to measure directly, one generally measure some quantity which is more or less proportional to it. Growth is therefore measured by the variety of the parameters, some of which are the increase in the fresh weight, dry weight, length, area, volume and the cell number. You may find it amazing to know that a one size maize root apical meristem can give rise to the more than the 17,500 new cells per hour, whereas the cell in the watermelon may increase in the size by up to the 3,50,000 times. In the former, growth is expressed as an increase in the cell number. The latter express the growth as an increase in the size of the cell. While the growth of the pollen tube is measured in terms of its length, an increase in the surface area denoted the growth in a dorsiventral leaf. Now the 15.1.3 is a phase of the growth. The period of the growth is generally divided into the three phases, namely the meristematic, elongation and the maturation. Let us understand this by looking at the root tip. The constantly dividing cell both at the root apex and the shoot apex represent the meristematic phase of the growth. The cell in this region are the rich in the protoplasm, possesses the large conspicuous nuclei. Their cell primary in the nature, thin and the cellulosic with abundant plasmodesmata connections. The cell proximal just next or away from the tip to the meristematic zone represent the phase of elongation. Increase the vacuolation, cell elongation and the new cell wall deposition are the characteristic of the cells in these phases. Further away from the apex, that is the more approximal to the phase of the elongation, lies the proportion of the axis which is undergoing the phase of the maturation. The cell of this zone attain their maximal size in terms of the wall thickening and the protoplasmic modification. Most of the tissues and the cell types you have studied in the chapter represent this phase. Now the 15.1.4 is a growth rate. The increase the growth per unit time is termed as a growth rate, and thus the rate of the growth can be expressed mathematically. An organism or a part of the organism can produce the more cells in a variety of the ways. The first is the arithmetic and second is the geometric. The growth are shown, and third is the zygotic divides. The growth rate shown as the increase that may be arithmetic or geometric. In arithmetic, growth follow the mitotic cell division and only one daughter cell continue to divide while the other differentiate and mature. The simplest expression of the arithmetic growth is exemplified by the root elongating at the constant rate. looking at the figure, on plotting the length of the organ against the time, the linear curve is obtained, that is the mathematically it is expressed as LT is equal to L0 plus RT. The LT is equal to length at the time t and L0 is the length at time 0. R is a growth rate per or elongate per unit time. Let us now see what happens in the geometric growth. In most systems, the initial growth is a slow, that is the lag phase, and it increases rapidly thereafter at an exponential rate, that is the log or the exponential phase. Here both progeny cells follow the mitotic cell division and retains the ability to divide and continue to do so. However, with limited nutrient supply, the growth slow down, leading to the stationary phase. If we plot the parameter of the growth against the time, we get typically the sigmoid or the S curve. The sigmoid curve is a characteristic of the living organism growing in a natural environment. It is typically for all cells, tissues and the organs of the plants. Can you think of more similar examples? What kind of the curve can you expect in a tree showing a seasonal activities? The exponential growth can be expressed as a W1 is equal to W0 ART. The W1 is a final weight, that is the weight, height, number, etc. and W0 is the initial size of the beginning of the period. R is a growth rate, T is the time of growth, E is the base of the natural logarithm. Here R is the relative growth rate and is also measure of the ability of the plant to produce the new plant material, refers to the efficiency index. Hence the final size of the W1 depends on the initial size of the W0. The quantitative comparison between the growth of the living system can also be made in two ways. The measurement, first is a measurement and the comparison of the total growth per unit time is called as the absolute growth rate. The second is a growth of the giving system per unit time expressed on the common basis. For example, the per unit initial parameter is called as a relative growth rate. In figure, the two leaves A and the B draw that are the different size but shows the absolute increase in the area in a given time to give the leaves A1 and the B1. However, one of them shows much higher relative growth rate. Which one and why? The 15.1.5 is a condition for the growth. Why do you not try to write down what you think are the necessary conditions for growth? This list may have the water, oxygen, nutrient as a very essential element for the growth. The plant cell grow in a size by a cell enlargement, which in turn requires the water. Turgidity of the cell helps in the extension growth, and thus the plant growth and the further development is intimately linked to the water status of the plant. Water also provides the medium for the enzymatic activities needed for the growth. The oxygen help in the releasing the metabolic energy essential for the growth activities. Nutrients and the macro and the micro essential elements are the required by the plants for the synthesis of the protoplasm and act as a source of energy. In addition, every plant organism has the optimum temperature range based study for its growth. Any deviation from this range could be detrimental to its survival. The environmental signals such as the light and the gravity also affects certain phases or a stage of the growth. Now the 15.2 is the differentiation, dedifferentiation and the redifferentiation. Cell derived from the root apical and the shoot apical meristem and the cambium differentiate and the mature to perform the specific function. This act leading to the maturation is termed as a differentiation. During differentiation, cell undergo the few or the major structural changes both in their cell wall and the protoplasm. For example, it forms a tertiary element. The cell would loses their protoplasm. They also develop a very strong elastic lignocellulosic secondary wall to carry the water to the long distance even under extreme conditions. Try to correlate the various anatomical features you encounter in the plants and the functions they perform. The plant shows the another interesting phenomenon. The living differentiated cell that by now have lost the capacity to divide can regain the capacity to divide and under certain conditions. This phenomenon is a term as a de-differentiation. For example, the formation of the meristem, interfascicular cambium and the cork cambium from fully differentiated parenchyma cells. While doing so, such a meristem or the tissues are able to divide and produce the cells that once again lose the capacity to divide but mature to perform the specific function, that is get the redifferentiation. List some of the tissues in the woody dicotyledonous plants that are the products of the redifferentiation. How would you describe the tumor? What would you call the parenchyma cells that are made to divide under the controlled laboratory condition during the plant tissue culture? Recall in the section of the 15.1.1 that is we have mentioned that a growth in a plant is open, that is it can be indeterminate or determinate. Now we may say that even a differentiation in a plant is open because the cell or the tissue arising out of the cell meristem and have the different structures at maturity. The final structure at a maturity of the cell or the tissue is also determined by the location of the cell within, and for example cell position away from the root apical meristem differentiate as a root cap cell, while those push to the periphery mature as a epidermis. Can you add a few more examples of the open differentiation correlating the position of the cell to its position in its organ? The 15.3 is a development. Development is a term that includes the all changes that an organism goes through during its life cycle, from the germination of the seed to the senescence. The diagrammatic representation of the sequence of the processes which constitute the development of the cell of the higher plant is given in a figure. That is, it is also applicable to the tissue or the organs. Plant follows the different pathways in response to the environment or the phases of the life to form the different kinds of the structures. This ability is called as a plasticity. For example, the heterophyly in cotton, then the coriander and the larkspur, etc. In such a plant, the leaves of the juvenile plants are the different in the shape from those in a mature plant. On the other hand, the differences in the shape of the leaves produces in the air and those produced in the water in a buttercup also represent the heterophylous development due to the environment. This phenomenon of the heterophyly is an example of the plasticity. Thus, the growth, differentiation and the development are also closely related events in a life of the plant. Broadly, development is considered as a sum of the growth and the differentiation. Development in the plant, that is both the growth and the differentiation, is under the control of the intrinsic or the extrinsic factor. The former includes both the intracellular, that is the genetics, or the intercellular factors, that is the chemicals such as the plant growth regulator, while the latter includes the light, temperature, water, oxygen and the nutrient, etc. Now the 15.4 is a plant growth regulators. Now the 15.4.1 is a characteristic. The plant growth regulators, that is the PGRs, are the small simple molecule are diverse chemical composition. They could be indole compound, that is the indole 3 acetic acid, that is the IAA, adenine derivative, that is the N6, furfuryl amino purine, that is the kinetin, derivatives of the carotenoids, that is the abscisic acid, that is the ABA, terpenes like the gibberellins, and the GA3 or the gases like the ethylene, that is the C2H4. The plant growth regulators are the variously described as a plant growth substances or the plant hormones or the phytohormone in a literature. The PGR means the plant growth regulator can be broadly divided into the two groups based on their function of the living plant body. The one group of the plant growth regulators are involved in the growth promoting activities such as the cell division, cell elongation, pattern formation, tropical growth, flowering, fruiting, seed forming, and these are also called as a plant growth promoter. For example, the auxins, gibberellins and cytokinin. The plant growth regulator of the other groups play an important role in the plant responses. It would and stress of the biotic and abiotic origin. They are also involved in the various growth inhibiting activities such as the dormancy and the abscission. The plant growth regulator abscisic acid belongs to this group. The gaseous plant growth regulator or ethylene could fit either of the group, but it is largely an inhibitor of the growth activities. The 15.4.2 is a discovery of the plant growth regulator. Interestingly, the discovery of each of the 5 major groups of the plant growth regulators have been the accidental. All these started with the observation of the Charles Darwin and his son is the Francis Darwin, when they observed that the coleoptile of the canary grass responded to the unilateral illumination by the growing towards the light resources, that is the photoperiodism. After the series of the experiment, it was concluded that the tip of the coleoptile was the site of the transmissible influences that causes the bending of the entire coleoptile. An auxin was isolated by the FW went from the tip of the coleoptile of the oat seedling. The Bacane, that is the foolish seedling disease of the rice seedling, was caused by the fungus pathogen, that is the Gibberella fusariae. The E. Kurosawa in 1926 reported the appearance of the symptoms of the diseases in a rice seedling when they were treated with a sterile filtrate of the fungus. The active substances were later identified as a gibberellic acid. The F. Skoog and his co-worker observed that from the internodal segment of the tobacco stem and the callus, a mass of the undifferentiated cell or the proliferated only if in addition to the auxin and the nutrient medium was supplemented with one of the following: extract of the vascular tissues, yeast extract, coconut milk or the DNA. The Miller et al. In 1955 later identified the crystallized the cytokinin promoting active substances that they term the kinetin. During mid in 1960s, the 3 independent researchers reported the purification and the chemical characterization of the 3 different kinds of the inhibitor. The inhibitor B, abscission second, and the dormin. Later, all the 3 were provided to chemically identical. It was named as abscisic acid ABA and HH causing in 1910 is confirmed the release of the volatile substances from the ripened organs that are the hasten the ripening of the store unripened bananas. Later his volatile substances was identified as ethylene, the gaseous plant growth regulator. Let us study the sum of the physiological effects of these 5 categories of the plant growth regulator in a next section. In the 15.4.3 is the physiological effect of the plant growth regulator. The 15.4.3.1 is the auxin. Auxin from the Greek is auxin means to grow. Was first isolated from the human urine. The term auxin is applied to the indole 3-acetic acid, that is the IAA, to the other natural synthetic compounds having the certain growth regulating properties. They are generally produced by the growing apices of the stem and the root, and from where they migrate to the region of their action. The auxin, like the IAA and the indole butyric acid, that is the IBA, have been isolated from the plants, that is the NAA, that is the naphthalene acetic acid, and the 2,4-D, that is the 2,4-dichlorophenoxyacetic acid. are the synthetic auxins. All these auxins have been used extensively in the agriculture and the horticulture practices. They help to initiate the rooting in the stem cutting an application widely used for the plant propagation. The auxin promote the flowering. For example, in the pineapple, they help to prevent the fruit and the leaf drop at early stage, but promote the abscission of the older mature leaves and the fruits. In most higher plants, the growing apical buds inhibits the growth of the lateral or the axillary bud, and phenomenon called as apical dominance. Removal of the shoot tip, that is the depication, usually result in the growth of the lateral bud. It is widely applied in the tea plantation, hedge making. Can you explain why? The auxin also include the parthenocarpy, for example the tomato. They are the widely used as a herbicide. 2,4-D, the widely used to kill the dicotyledonous weeds, does not affect the mature monocotyledonous plant. It is used to prepare the weed-free lawn by the gardeners. The auxins are the control the xylem differentiation and helps in cell division. The 15.4.3.2, the gibberellin. Gibberellin are another kind of the promotory plant growth regulator. There are the more than the 100 gibberellin reported from the widely different organisms such as the fungi and the higher plants. They are denoted as a gibberellin 1, GA1, GA2, GA3 and so on. However, gibberellic acid, that is the GA3, was one of the first gibberellin to be discovered and remain the most intensively studied form. All gibberellic acids are the acidic. They produces the wide range of the physiological responses in plants. Their ability to cause an increase in the length of the axis is used to increase the length of the grape's stalk, and the gibberellins cause the fruit like apples to elongate and improve its shape. They also delays the senescence, and thus the fruit can be left on the tree longer so as to extend the market period. The gibberellic acid GA3 is used to speed up the malting process in brewing industries. The sugarcane store the carbohydrate as a sugar in their stems. The spraying sugarcane crop with a gibberellin increase the length of the stem and thus increasing the yield by as much as the 20 tons per acres. The spraying juvenile conifers with the gibberellic acid has turned the maturity period, thus the leading to the early seed production. Gibberellin also promote the bolting internode elongation just prior to the flowering in beet, cabbage, mini plant and the rosette habit. The 15.4.3.3 is a cytokinin. Cytokinin have the specific effect on the cytokines and were discovered was the kinetin, that is a modified form of the adenine, like a purine. From the autoclave herring the sperm, that is the DNA. The kinetin does not occur naturally in the plant. Search for the natural substances with the cytokinin like the activity led to the isolation of the zeatin from the corn kernel and the coconut milk. Since the discovery of the zeatin and the several naturally occurring cytokinin and some synthetic components with cell division promoting activity have been identified, and the natural cytokinin are synthesized in the region where the rapid cell division occurs. For example, the root apices, developing shoot buds, young fruit, etc. It helps to produce in new leaves, chloroplast in the leaves, lateral shoot growth and the adventitious shoot formation. Cytokinin helps to overcome the apical dominance. They promote the nutrient mobilization which help in the delay of the leaf senescence. The 15.4.3.4, that is the ethylene. Ethylene is a simple gaseous plant growth regulator. It is synthesized in the large amount by the tissue undergoing the senescence and ripening the fruit. The influence of the ethylene of the plant include the horizontal growth of the seedling, swelling of the axis and the apical hooks formation, in the dicot seedling. Ethylene promotes the senescence and the abscission of the plant organ, especially to leaves and the flowers. Ethylene is a highly effective in the fruit ripening. It enhances the respiration rate during the ripening of the fruits. This rises in the rate of the respiration is called as a respiratory climactic. Ethylene breaks the seed and the bud dormancy, initiate the germination in the peanut seed, sprouting of the potato tubers. Ethylene promotes the rapid internode and the petiole elongation in the deep water rice plant. It helps the leaves or the upper parts of the shoot to remain above the water. Ethylene also promotes the root growth and the root hair formation, thus helping the plants to increase the absorption surface. Ethylene is used to initiate the flowering and for the synchronizing the fruit set in the pineapple. It also includes the flowering in the mango. Since the ethylene regulates so very physiological processes, it is one of the most widely used plant growth regulator in the agriculture. The most widely used compound as a source of the ethylene is the ethethone. The ethethone is the aqueous solution is radially absorbed and transported within the plant and release the ethylene slowly. The ethethone, that is the hasten the fruit ripening in the tomato and the apple and accelerate the abscission in the flower and the fruit, that is the thinning of the cotton, cherry, walnut, etc. It promotes the female flower in the cucumber, thereby increasing the yield. Now the 15.4.3.5, that is the abscisic acid. As mentioned earlier, the abscisic acid, that is the ABA, was discovered for its role in the regulating the abscission and the dormancy. But like other plant growth regulator, it is also has the other wide range effective on the plant growth and the development. It acts as the general plant growth inhibitor and the inhibitor of the plant metabolism. ABA, that is the abscisic acid, inhibits the seed germination. Abscisic acid stimulates the closure of the stomata and increase the tolerance of the plant to various kinds of the stresses. Therefore, it is also called as a stress hormone. The abscisic acid plays an important role in a seed dormancy, maturation and the dormancy. By inducing the dormancy, abscisic acid helps the study to withstand the desiccation and other factors unfavorable for the growth. In most suitable, the abscisic acid acts as the antagonist to the gibberellic acid. We may summarize that for any and every phase of the growth, differentiation and the development of the plant, one or the other plant growth regulator has some role to play. Such role could be complementary or the antagonistic, and this could be individualistic or the synergistic. Similarly, there are the number of the events in the life of the plant where the more than the one plant growth regulator interact to affect the event. For example, the dormancy in the seed or the bird's abscission, senescence, apical dominance, etc. Remember the role of the plant growth regulator is of the only one kind of the intrinsic control, along with the genomic control and the extrinsic factors. They play an important role in the plant growth and the development. Many of the extrinsic factors such as the temperature, light, control of the plant growth and the development via the plant growth regulator. Some of such events could be the vernalization, flowering, dormancy, seed germination, plant movement, etc. We shall discuss briefly the role of the light and the temperature or both of them extrinsic factor on the limiting of the on initiation of the flower. The first, the 15.5 is the photoperiodism. It has been observed that some plants require a periodic exposure to the light to induce the flowering. It is also seen that such plants are able to measure the duration of the exposure to the light. For example, some plants require the exposure to the light for the period exceeding a well-defined critical duration. The other must expose to the light for the period less than this critical duration before the flowering is initiated in them. The formal growth of the group of the plants are called as a long day plant, while the latter one is termed as a short day plants. The critical duration is different for the different plants. There are the many plants, and however, where there is no such a correlation between the exposure of the light duration and induction of the flowering responses, such a plant are called as a day neutral plant. It is now also known that not only the duration of the light period but that of the duration of the dark period is also of equal importance. Hence it is said to be the flowering in a certain plant depend not only on the combination of the light and the dark exposure but also their relative duration. This response of the plant to the period of the day or the night is termed as a photoperiodism. It is also interesting to note that while the shoot apices modified themselves into the flowering apices prior to the flowering, that is the shoot apices of the plants by themselves cannot perceive the photoperiods. The site of the perception of the light or a dark duration are the leaves, and it has been the hypothesized that there are the hormonal substances that is the responsible for the flowering. The hormonal substances migrates from the leaves to the shoot apices for inducing the flowering only when the plants are exposed to the necessary productive photoperiods. Now the 15.6 is the vernalization. There are the plants for which the flowering is either quantitatively or the qualitatively depends on the exposure of the low temperature, and this phenomenon is termed as a vernalization. It prevents the precocious reproductive development late in a growing region. The enable plants to have the sufficient time to reach the maturity. Vernalization refers specially to the promotion of the flowering by the period of the low temperature. Some important food plants, wheat, barley, rye, have two kinds of the varieties, the winter and the spring variety. The spring variety are normally planted in the spring and come to the flower and produces the grain before the end of the growing season. Winter varieties, however, if the planted in the spring, would normally fail to flower or produce the mature grain within a span of the flowering season. Hence they are planted in autumn and they germinate and over a winter come out as a small seedling and resume the growth in the spring and are harvested usually around the midsummer. Another example of the vernalization is seen in the biennial plants. That is, the biennial are the monocarpic plants that normally flowers and die in the second season. That is, the sugar beet, cabbage, carrot are some of the common biennial plants. The subjecting the growth of the biennial plants to the cold treatment stimulates a subsequent photoperiodic flowering responses. The 15.7 is the seed dormancy. There are the certain seeds which fall to germinate even when the external conditions are favorable. Such seeds are understood to undergoing a period of the dormancy which is a control not by the external environment but are under endogenous control or a condition within the seed itself. Impermeable and the hard seed coat, presence of the chemical inhibitors such as the abscisic acid, the phenolic acid and the ascorbic acid and the immature embryos are some of the seasonal which causes the seed dormancy. The seed dormancy, however, can overcome through the natural means and various other man-made measures. For example, the seed coat barrier in some seeds can be broken by the mechanical abrasions and using the knives, that is knives and the sandpaper, etc., or though vigorously shaking. In the nature, these Aberrations are caused by the microbial action and passage through the digestive tract of the animals. Effect of the inhibitory substances can be removed by the subjecting the seeds to chilling conditions or by application of the certain chemicals like gibberellic acid and the nitrates. The changing the environmental conditions such as the light, temperature and are the other methods to overcome the seed dormancy. Now the summary of the plant growth and the development. Growth is one of the most conspicuous event in any living organism. It is an irreversible increase expressed in the parameters such as the size, area, length, height, volume, cell number, etc. It conspicuously involves the increase that is the protoplasmic material. In plants, the meristem are the site of the growth, the root and the shoot apical meristem, sometime along with the intercalary meristem, contributes to the elongation growth of the plant and axis. Growth is an intermediate in the higher plants. Following the cell division in the root and the shoot meristem cells, the growth could be arithmetic and the geometric. Growth may not be and the generally not sustained at the high rate throughout the life of the cell or tissues or organs or organism. One can define the three principal phases of the growth: the lag, log and senescent phase. When a cell loses the capacity to divide, it leads to differentiation. Differentiation results in the development of the structures that is the commensurate with the function and the cell finally has performed. Generally, the principle of the differentiation of the cell, tissue and the organs are the similar. The differentiated cell may differentiate and then re-differentiate. Since the differentiation in the plant is open and the development could also be flexible, it develops in the development is in the sum of the growth and the differentiation. The plant exhibit the plasticity in the development. The plant growth and the development are under control of the both intrinsic and the extrinsic factor. Intercellular or intrinsic factors are the chemical substances called as a plant growth regulator, that is the PGR. There are the diverse group of the PGR, that is a plant growth regulator in the plant, and principally belonging to have the growth auxins, gibberellin, cytokinin, abscisic acid and the ethylene. These plant growth regulators are the synthesized in the various parts of the plant. They control the different differentiation and the developmental events. Any plant growth regulator has their diverse physiological effect on the plants. The diverse plant growth regulator are the manifest similar effects. The plant growth regulator may act the synergistically or the antagonistically. The plant growth and the development is also affected by the light, temperature, nutrition, oxygen status, gravity and such an external factor. The flowering in some plants is induced only when exposed to the certain duration of the photoperiod. Depending on the nature of the photoperiod requirement, the plants are called as a short day plant, long day plant, day neutral plant. Certain plants also need the exposure to the low temperature so as to hasten the flowering later in the life, and this treatment is known as the vernalization. Thank you so much.